All right, proteins. Part 4. They have many diverse functions in the body. They can serve, their enzymes act as catalysts, speeding up metabolic reactions. They defend you. They transport things like hemoglobin. They can contribute to structure support of your body, cause movement, regulate things in your body, perform storage. Important. So, to build one, you need to assemble the monomers called amino acids. There's 20 different types of amino acids found in living things. Every amino acid has an amine group with a nitrogen and a carboxyl, COOH, and they're covalently linked to the center carbon. And then that carbon can be covalently linked to what's called a functional group called an R group, and then a hydrogen. That R group is what makes different amino acids different. So here's a monomer, here's an amino acid, here's an amino acid, okay? They are covalently linked by a bond called a peptide bond. To put two of them together, you need to have a dehydration, here's this word again, synthesis reaction. So to produce protein, many dehydration synthesis reaction produces it in many water molecules as a byproduct. Because to form, put two things together, we need to lose an H from one and an OH from the other. Here is one amino acid. Here's two of those being put together in the water that's produced. And here is a polymer. At one end of a protein, there's always an amine group, an N with two hydrogens. I think I said 3 in another video. I meant 2. And then at the other end is the carboxyl group, the C double bonded to an oxygen with an OH. Notice all the R's are called R groups. Okay, so amino acids, if we had 3 or more bonded together, it has a special name called an oligopeptide. If there's above 21, it's called a polypeptide. And a protein is actually 200 or more amino acids put together. We also have these things called glycoproteins, which have a carbohydrate group part of it. The blood types, A, B, and O. The blood types are determined by these glycoproteins on the surface of the red blood cells. And we have now talked about the 4 major types of macromolecules. Monomers, the monomers of proteins are amino acids, and the bond is a peptide bond. And this is just showing images of the 4 macromolecules. Here's our fats. We see the triglycerides, we see the phospholipids, the steroids, okay, and their functions. Carbohydrates, here we see a monomer, glycogen, how it can go back and forth, how glycogen is stored in the liver. We need carbohydrates for energy. Nucleic acids, we see RNA and DNA inside the nucleus. Here is the nucleotides that you use to make them. And then the proteins, here's one amino acids that are linked together to form large proteins that have many different roles in the body. And here are the 20 different types of amino acids. We see in white here how they all have a different white region. That's the R group. That's how they differ. Everything else about the amino acid is exactly the same. They differ by that R group. Okay, so next, something called protein conformation, or the building of a protein. To build a protein, the first level, the simplest level of structure is called the primary structure. All it is, if you were to put beads on a chain right now, you would do the primary structure. Beads on a chain is the series of amino acids in order. Now, taking that primary structure and making it more complex is called conformation. It gives a 3-dimensional shape to the protein. The arrangement or the folding beyond the primary structure involves intermolecular attractions. So we're talking van der Waals forces, we're talking hydrogen bonding and the like. So hydrophobic interactions are the nonpolar interactions, van der Waals, and the polar interactions are going to be hydrogen bonds between our groups, okay? And we also see ionic bonds in there and other types of specific bonds, okay? So primary structure is the amino acids linked together. Secondary structure is in two forms, either what's called an alpha helix, so like think of a curling iron to curl somebody's hair, and then a beta sheet if you were to fold a paper back and forth on top of each other to create a fan. And then a tertiary structure is a 3D shape given to that molecule. So it's level up, a little bit more folding, a little bit more chemical bonding between different parts of the molecule. There are two categories: globular, forms into a compact shape, and then fibrous is just that linear molecule, that helix, just folded even more. The quaternary structure is the top level of complexity, and instead of just one protein chain we're talking about, or amino acid chain, we're talking about more than one. So this level is only present in proteins that have two or more chains of amino acids. Hemoglobin would be a great example because it has 1, 2, 3, 4 different amino acid chains put together to make that protein. Sometimes we have non-protein groups hooked to that protein that are covalently bonded. This is called a prosthetic group. There's an example with hemoglobin. Denaturation is something that happens with proteins when their chemical bonding is disrupted somehow. So there's a conformational, that structure, it changes somehow in that protein. Usually that change is irreversible, and it will change how that protein works. Usually if a protein is denatured in the body, it's not a good thing. And high temperature can cause proteins to denature, or changes in pH. And this just goes back to high temperatures are dangerous. You don't want a person's temperature to get dangerously high, because proteins that make up your body's structure and how your bodies function can denature or change chemically, so they can't do the things they do. Same with large swings in pH, swings. And this just shows actually how the chemical bonds break if there's a increased hydrogen amount, which means a decreased pH, or the opposite. So alkalosis down here, or acidosis. And it can be lethal. Changing the protein structures in your body can be lethal. That's called denaturation. So distinguish between the primary, secondary, tertiary, and quaternary. Make sure you review. It's a level of complexity that increases. There's different foldings, shapes, and different amounts of protein chains that depend on the level of complexity.